Hi, I'm Bethanie Morrison from the laboratory of Dr. Mary Cutler in the Department of Pathology at the Uniformed Services University of the Health Sciences. Today, we will be showing you a procedure for the development of mammospheres from HC11 mouse mammary epithelial cells. We use this procedure in our lab to study lactogenic differentiation. So let's get started.
To start this protocol, HC11 cells are grown to confluence for 6 days in rPMI 1640 medium, which is supplemented with 10% fetal calf serum, 5 micrograms per milliliter insulin, 10 micromolar HEPs, and 10 nanograms per milliliter epidermal growth factor, EGF, to establish competence. These cells have been used extensively by the laboratories of Nancy Hines and Bernard Groner to study the regulation of lactogenic differentiation. In the tissue culture hood, the cells are then washed with PBS and maintained in growth media without EGF for 24 hours.